Great, thank you. Yeah, so my name is Katherine Silliman. I'm currently a marine scientist with the South Carolina Department of Natural Resources. And I'm really excited to talk to you all today about the MarineOmics Working Group and what we've been up to for the last year and a half or so. In particular, a new web resource we've developed for robust and reproducible genomic research in marine and aquatic science. I really wish I could be there this week to meet with everyone, but I have a very small baby at home. So also if I'm a little tired and not firing on all cylinders today, I apologize. But fortunately, my co-organizer and co-leader of the working group, Sam Bogan, is there. So if anything from this talk really interests you or excites you, make sure you reach out and connect with him. And so before I get started, I really want to highlight the other members of the working group who put a lot of time and energy into what we've produced. So including Sam, there's Alan Downey Wall, Chris Mantegna, Danny Davenport, Hanny Rivera, Jesse Clinkham, Joanna Griffiths, Joe McGear, Caitlin Macarenas, Megan Guidry, Natalie Merrill, Nicholas Liu, Sarah Shaw, Sarah Wuchik, and our faculty mentors, John Puritz, Stephen Roberts, and Andrew Whitehead. And of course, funding that we received through NSF with the RCN. And so today I'm going to just talk about who the MarineOmics Group is, what we did, and a little bit about how we did it. So, thinking about how to synthesize existing bioinformatic resources and identifying gaps in the field. And then what's next for the MarineOmics Working Group and the website, such as future topics we'd like to include, as well as how to get more community involvement so this resource can be useful and persist into the future. So the MarineOmics Working Group idea started when I was a postdoc at Auburn, and I was really excited about going to the RCN Genomic Workshop in August 2020, which, as we all know, was canceled. And part of why I was excited about this workshop is because I wanted a chance to go and talk to other experts in the field to maybe come up with some consensus on the right way to do bioinformatic and genomic analyses on non-model species. And this came out of my graduate experience where it really seemed like the Wild West. There wasn't a lot out there on the right way or the best way to apply methods that had been used a lot more in humans or Drosophila to weird marine species whose genomes maybe weren't in the same great shape as those model species. But when the workshop was canceled and the RCN put out a call for these working groups, I thought this might be a great opportunity to bring some of those folks together. And at the same time, Sam Bogan, a PhD student at UC Santa Barbara, was also interested in starting a working group. So we combined forces and made a proposal with the mission statement of promoting rigorous and reproducible genomic research on non-model species, especially in marine and aquatic science. And so there's a lot of ways people define rigorous and reproducible. Here, where our definition of rigorous is following best principles to identify and mitigate the sources of error that occur in data generation and analysis. And there's actually a great example of the importance of that from Katie Lauderhouse's group. Oops, sorry, I have a little bit of feedback. That's okay, I'll push through. A great example out of Katie Lauderhouse's group, where here is showing the use of different outlier tests for genetic differentiation through time with the number of citations on the y-axis. And the vertical lines are showing evaluation studies published that demonstrated high false positive rates for two of these methods, FDIST and Losartan, which are shown in the dotted lines. And as we would hope, after evaluation studies say, maybe these aren't the most rigorous tests to use, we'd hope to see their use decline. And we do see that, but there's still, even 10 years later, a fair amount of studies using these methods. And so sort of highlighting the importance of both putting out evaluations like this, but also promoting them in a way so that members of the community see them and start to change sort of what their approaches are. And then with reproducible, here I mean when others can reproduce the results of a study using accessible data, code, and documentation. And this is something that's become really important to me throughout my career, and it's also really, really hard to do. I certainly don't meet this goal every time. But as we saw from Rachel's talk yesterday, having better reproducibility and data management is really important for synthesis across studies now and in the future. And so we started our work in December 2020. At different times, we've had 18 members across 13 institutions, undergrads up to our faculty mentors. And we also were able to broaden participation in the field with funding from the RCN for internships, which I'll talk about in a little bit. And we have expertise on population genomics, epigenomics, transcriptomics, and our research backgrounds span a wide type of study species, which I think has really helped us come at these topics and issues with different backgrounds and perspectives. So early on in our discussions, I made a comment about wanting to develop some best practices for genomic methods in non-model species. And our faculty mentor, John Puritz, said, you know, I don't really like the term best practices. I prefer best principles. And so if you know John, or at least follow him on Twitter, you know he really likes memes. So I had to turn him into a meme. But the idea of best principles is that every data set is different. So best practices sort of elicits this idea of there's one right way to do an analysis. But actually, it's better to think about, instead of a rigid set of parameters or ways to filter your data, we want to encourage data exploration and rigorous quality control. And so this idea of best principles has been sort of a guiding concept throughout the work that we've done. And so synthesizing anything in the internet age is really hard. And the same is for the vast amount of bioinformatic resources that are out there. So I wanted to talk a little bit about our process for tackling this. We started out with a survey of RCN members to identify what the priorities were in the community in terms of what topics they'd like to see more guidance on. And then we discussed results of that survey in the context of our working group strengths to set priorities that were achievable and that we thought that we could contribute best to. We then, within those, brainstormed specific topics that we thought were in need of more guidance. But after that, we then looked hard for what resources existed out there. You know, in some cases, maybe it's just hard to find the guidance that exists. And we drew from review papers, method evaluation papers, comparative studies, so an empirical study that uses two different methods, and talked about, you know, how those methods varied for their study, tutorials, blog posts, and even GitHub repositories from papers that do a really good job of reproducibility. We then hosted expert panels on different topics to try to find areas that experts had some consensus on, or maybe were more debated in the field, or there really didn't exist the empirical basis to make strong decisions yet. And then finally, I wanted to mention, you know, I'm sure we're all a lot better at remote collaboration at this point, but certainly this was something that was really new to me, working with a working group of this size remotely. Most of us have never met in person. I still haven't met Sam in person, though we've spent many, many Zoom hours together. And so just some of the things that worked for us in this setting was, one, co-leadership. You know, Sam and I brought very different skills to the working group. And I think that was crucial to getting the job done. And also if someone was busy, someone else could pick up the slack and trade back and forth. We always tried to have meeting agendas before working group meetings, and this helped us, I think, be really efficient with our time. And then we tried to incorporate some active learning techniques, if you've heard of these from classroom settings, to try to really engage everyone, even though we were on Zoom, and give everyone a chance to contribute and make their thoughts known. And then finally, just assigning action items with at least some rough deadline of when to be done, I think helped us move things forward. So as I mentioned, one of our first activities were these virtual panel seminars in May and June of last year. And so we had one on genotyping with whole genome sequencing. We had one on reduced representation sequencing, such as RAD-seq. We had one on population genomic analyses, such as structure or identifying outliers, and then one on RNA-seq and differential gene expression. So these are linked to on the MarineOmics site and on YouTube, and I highly recommend checking them out if you haven't yet. There's a lot of great information and perspectives in there. We then hosted 3 virtual undergraduate research interns in the spring of last year with funding from the RCN. These research interns, in addition to mentorship and some skill development, played a really crucial role in one of our other main products, which is a literature review of reproducibility in marine studies of adaptation that use omics methods. And so by literature review of reproducibility, we really didn't want it to just be a, here's all the ways that reproducibility is bad in these kind of studies. We mostly want it to be a chance to highlight studies that are doing things really well, draw some best principles from our observations, and also highlight barriers or catalysts that may exist in reproducibility for synthesis, much like Rachel was talking about yesterday. And so just a little highlight, this is from 223 studies that we have showing code availability through time. And so as we would hope, a lot more code is available in recent studies, but there's still many, many studies that don't have any available code. And so that's one area that can be improved on. And then finally, our magnum opus, I guess, is this our MarineOmics website, which is a dynamic web resource with guidelines geared towards both people new to studies, genomic analyses, as well as experienced users. And we really want this to be a place to foster dialogue and host content for evaluating and applying ecological genomics methods. So we do this through guidelines, in-depth code tutorials, literature summaries, as well as some links to the many resources that already exist out there. And one note about our code tutorials is we make a concentrated effort to provide justification for parameter choices that are made when you're going through a bioinformatics pipeline, as well as some tips to avoid common pitfalls. So the current topics on the website include information to help choose a population genomics approach based on your study and your questions. We have more directed content for genotyping and data filtering for reduced representation sequencing, such as RAD-seq, whole genome resequencing, both low and high coverage, and pooled sequencing. And then in development, we have work on population genomic analyses, both with hard-called genotypes and genotype likelihoods, like what you would get from low coverage whole genome sequencing. And then we have a functional genomics section with RNA-seq. And this section was developed, spearheaded by Sam Bogan, and it's looking at how to apply existing differential gene expression packages for experiments that have multiple factors and are interested in interaction terms. And this is one of the areas that we started with because many marine studies of adaptation are increasingly using multiple environmental or biotic factors. However, there's really not a lot of guidance out there on how to apply existing differential gene expression packages towards these complex model setups. And so just like a little example from that section, here is a correlation matrix of p-values obtained for interaction terms from Lima-Voom, EDGE-R, and DSeq2, which are popular differential gene expression packages. And you can see there's not a lot of correlation. And depending on what package you choose, you might get a different set of significant genes. Something else that we highlight on the website is the utility of PCA for data exploration. So here on the left is a PCA where the individuals are colored by where they were collected. But we see two outliers from two different sites really driving this PCA. And so the importance of identifying and investigating outliers like this before you move on to population genomic analyses. And it turns out these were likely cryptic species. And so we definitely don't want to be throwing them into population genomic analyses without taking that into account. Something else we talk about a lot is sort of the non-experimental sources of variation that you might get, such as from different sequencing batches. So here in panel A is a PCA where individuals are colored by the sequencing batch that they were prepared in. And we see strong clustering by sequencing batch, which hides the variation based on just actual population genetics. And so we talk about ways to test for these batch effects and then remove them. So you get a PCA like in panel B, which is no longer structured by the sequencing batch. And so the next big step for the website is more community engagement, which is why I'm excited to talk to folks here about the website. We really want this to be open sourced. And so that's why the website is hosted through GitHub. And we also have a GitHub discussion section linked on the website. And here is a place we want people to post ideas for new content they'd like to see, ways the website could be extended, or if you read something and you have a correction or a disagreement or a reference you think should be added, a place for that sort of peer review as well. And since it's on GitHub, if you want to write new content or make edits to content, you can use GitHub pull requests to do that directly. Everything is written in R Markdown, which just knits to an HTML. And then if you are really excited about this work and want to contribute in a more formal way, we'd love for more folks to join the MarineOmics group. And Sam and I can talk about all the ways that you can participate in that way. And then another use that I'd love to see the website used for is to make it sort of a community discussion forum. So a place where folks can go to request as well as provide project-specific advice. One of the cool things that came out of our panel seminars was folks would say, I have this kind of data, it looks like this. What do you think I should do as my next steps? And while there are BioStars and Stack Overflow for software specific questions, it feels like there's not really a great forum for project specific advice in non-model species. So we think that this could be a great place to get that going. And then some of the future topics that we are planning to integrate. One is how to integrate multiple omics data types, which I think there's a panel on later today. And is an area of huge interest in the RCN. More general guidelines on how to assess and compare different population genomic analyses. For functional genomics, we'd like to have a primer on ATAC-seq for non-model species, as well as how to integrate that with RNA-seq data and comparison of functional enrichment methods. And then we are really lacking in a section on genome assembly and annotation, in part because that didn't reflect the expertise we had in our original working group members. And so if that's a space that you've worked in a lot or have a lot of thoughts on how to do in a rigorous manner, please reach out to us. We would love for you to contribute to the website. And then, of course, with our reproducibility literature review, distilling some best principles for improving reproducibility in the field. And with that, I want to just acknowledge again all the work by the MarineOmics Working Group and our funding. And with that, I'll take any questions.
Great. Do we have any questions?
I have a quick question. And yeah, I have a lot of questions and I'm excited to chat further because we have a lot in common, I think. But I'm curious if you happen to look at the timing of the journals that were, if the journal itself is requiring code or not. You had that graph about how many papers had code in them. And I'm curious how it matches up with the journals, and something that we thought a lot about with our kind of metadata work was basically like how much of it should be top down of like a journal or some institute mandating it versus how much of it is bottom up of like just asking researchers to do a good job. So I'm curious if you looked at that or have plans to.
Yeah, we have metadata on what all the journals are, and that's something that we're going to be explicitly investigating. Is are there some journals that have better or worse because of their policies? But yeah, I don't have the exact results from that yet, but that's definitely something that we're tying into this. Yeah, that's a great point.
Cool, thanks.
Great. Yeah, and I guess we couldn't see here on the screen, but that was a question from Rachel, right?
Oh
yes,
yeah, sorry.
Sorry, we couldn't. It didn't switch on our Zoom screen here, but great. Next, Diana has a question.
Yeah, so I love the idea of having sort of a place to communicate questions or almost network about more project specific questions. And I'm wondering about whether that's kind of linked to those individuals' GitHub pages, because you almost want to be able to, I think one of the things I really dislike about some of the existing resources, particularly, BioStars or those sorts of things, is you don't know actually what that person's expertise is. So it's a little bit of the blind following the blind. So
yeah,
if it links to that person's GitHub page, you can look at some repositories and see very clearly, oh, they have 5 projects that use this approach. This is definitely in their wheelhouse. That type of thing.
Yeah, I just made a note because I think that's a really a cool thing to ensure. I'm pretty sure you have to have a GitHub account to comment on the discussion, and so in that way it would link to it. But not everyone is actively using GitHub yet, but they might have that expertise. So maybe in our guidelines for use, saying if you don't have an active GitHub, make a comment about this is where my expertise is. This is my Google Scholar or something like that. So it's linked in a better way. Yeah, great comment.
And then that's going to be the final question or comment we have time for.
Just responding briefly to the previous question about journals and code requirements. So putting on my hat as editor-in-chief of a journal, we started back in January of this year requiring code for submission. So data and code both have to be available on submission. You can use data Dryad, for example, and create a private key. So it's not public yet. It's available just to the reviewers. We know who the reviewers are. So if somebody publishes your data before you do, we know that, and we know who had access. And so, that's pretty tightly controlled that way. And I know there are a couple of other journals that are considering code requirements. The other thing that we started doing a year ago is we now have data editors whose job is to check your data repositories and confirm that it contains what it's supposed to contain. The code is annotated. The metadata is clear. The README file tells us what units your variables are and what your column names mean and so on. There are a few other journals that are moving that way, but at the moment, like code and checking repositories, very few journals do. And so it's mostly just a trust system. We're believing if you say you archived your data, we don't even look. We just say, okay, great. You said you did.
Add to that, JGR Oceans and most of the AGU journals are moving towards code and data having to be published. And if you ask, the reviewers are asked and have to check off on the review form that they've checked that those exist. Whoever downloads it and makes sure it works, that's probably less likely.
Yeah, that's actually a question I had for Dan. Do you have a sense for how many reviewers are actually using the code or data looking at it?
We can get that data because it is documented in our system. So I'll ask the journal. What I will say is that about 1 in 10 papers that I write a decision on, some reviewer has gone and redone analyses. They've like rerun something. They've been looking at the code enough to say, hey, I reran your general linear model binomial and it's returning a warning that it's failing to converge. What's up with that? So I'd say about at least 10% are finding a problem in the data or code that they are opening up and analyzing themselves. It's probably not much more than that, frankly, because it is a lot of extra work. It's not something that traditionally one requires of reviewers.
I just want to read the comments that are happening on Zoom. So John Purritt said there's a big difference between code and reproducible code, which is true. And Katherine said, yes, 100%. We are evaluating code quality in our review as well, and it varies a lot. And then Rachel previously talking about how to know who is giving you advice on the GitHub said perhaps it could be linked to a ResearchGate profile so people can investigate the advice they're getting about how to handle their data and analyses. Thank you so much. Let's thank Katherine again. So next up we have Savannah Lepura. Are you there, Savannah? Anyone on Zoom? Do we have Savannah? Okay. Do you want to just go ahead with the discussion, Katie? Or should we try to reach her? Or what do you think? Yeah.
Okay, so I guess we're just going to rearrange the schedule a little bit. Just give me a second here to set up my screen.